thank you for invitation. Hello from snowy Poland. I hope that you can hear me, and I would like to put on the presentation now. Okay. Yeah, can you see the screen now? Yes? Okay.
Yes, we can see.
Okay. I would like to briefly present the advances in clinical trials in non-Duchenne muscular dystrophies. Why non-Duchenne? Because presenting all the clinical trials in Duchenne muscular dystrophies will take the whole lecture, another lecture. I'm happy that Dr. Lehmann already presented briefly the state of clinical trials in Duchenne muscular dystrophy. Of course, Duchenne muscular dystrophy is the most frequent one, but therefore I would like today to focus on all the other ones. Okay, first of all, this is my disclosure. The source of information I based on generally available information, like, for example, the site clinical trials and trial sponsors publications. I will limit, due to the limited time, only to pharmacological trials, only to drug trials. First of all, I am a clinician and I am not currently engaged in any of the presented clinical trials. I just did this review predominantly to present it to the patients community, to have the information gathered in one place for them. We can divide muscular dystrophies into different types. First are dystrophinopathies. These include Duchenne and Becker muscular dystrophy, limb-girdle muscular dystrophies, which overlap in some percentage with congenital muscular dystrophies. Another type is facioscapulohumeral muscular dystrophy, myotonic dystrophies type 1 and 2, Emery-Dreifuss muscular dystrophy, and oculopharyngeal muscular dystrophy. And currently, interventional clinical trials, pharmacological trials are going in the of dystrophies, which I highlight as well, but I will skip limb-girdle muscular dystrophies. And I will talk today about these types of dystrophies. First of all, we already know that steroid can improve the patient's condition, can slow the progress of the disease in Duchenne muscular dystrophy, but we still don't have any guidelines concerning other types of muscular dystrophies, whether it is beneficial to patients to use steroids in other types of muscular dystrophies. So there are some trials which are ongoing in Becker muscular dystrophies and limb-girdle muscular dystrophies. In Chicago, in USA, there is a trial of weekly prednisone dosing. This is ongoing. This study has not published results yet. There was a study of deflazacort use in limb-girdle dystrophy type 1. However, it was terminated early, unfortunately, because this is one of the dystrophies which, according to some data, could benefit from the steroid use. It was terminated early due to high dropout, unfortunately. There is another study going in Becker muscular dystrophy with vamorolone. This is currently recruiting the patient in USA. Vamorolone is called a dissociative steroid. It is a steroid derivative which has retained anti-inflammatory activity, but doesn't have GRA-related transactivations, so it doesn't have many corticosteroid side effects. For example, does not present mineralocorticoid activity. Yeah, so this is the safest alternative, and it should retain the anti-inflammatory activity of steroids. Okay. Apart from the trials with steroids, there are some very interesting new substances tested. One of them is EDGG 5506 by Edgewise Therapeutics. It is a selective fast skeletal muscle myosin inhibitor. Myosin, it is a protein which is present in the muscle and which is responsible for the contraction of the muscle. And fast fibers, we have fast and slow myosin types, and fast fibers are more susceptible to injury due to muscle contraction related stress. This injury due to muscle contraction related stress is especially important, is especially enhanced in muscular dystrophies, in different types of muscular dystrophies. And in mouse dystrophinopathy model, selective inhibition of fast fiber contractions prevents this type of injury. So the sponsor designed a trial in which the substance is blocking the fast skeletal muscle myosin. There was a preliminary study called ARCH in patients with Becker muscular dystrophy. This study is still ongoing, but has the interim data after 6 months. And we can see this is the North Star ambulatory scale, which assess the ambulation. The higher, the better score. And this is the natural Becker muscular dystrophy history, and this is the score, the progress of the score in the Becker muscular dystrophy patients treated by this EDGG 5506. And this is the creatine kinase level, which is decreased by about 40% after 6 months. So these are very encouraging preliminary data, and the company has started already 2 other trials. Canyon trial in adults and adolescents with Becker muscular dystrophy, which is currently recruiting patients. Lynx trial in children with Duchenne muscular dystrophy. And another very interesting trial is planned in patients with Becker, but also limb-girdle muscular dystrophy, 2I, this is type R9 currently, McArdle myopathy. Yeah, so this is planned soon to start. Another group of clinical trials are trials with gene therapies, and I collected them in form of the table. There were some trials of gene therapy in dyspherinopathy, in limb-girdle dystrophy type 2. The very preliminary phase 1, this trial is completed, but the results are still pending. We don't know the results. There was also a study in this sarcoglycanopathy type alpha, LGMDR type 3, which also has been completed, and it showed the increased sarcoglycan production and local muscle function improvement in the injected limb. This was a single limb injection. Another trials are currently active is gene therapy in sarcoglycanopathy beta LGMDR4, which is also a preliminary phase clinical trial is ongoing but not recruiting patients. And now a very interesting clinical trial with gene therapy in limb-girdle muscular dystrophy type R9, fukutin-related protein related. This is quite a common type of limb-girdle muscular dystrophy. And currently the study is recruiting patients in Denmark, France, and then UK. Another very similar study is planned, but not yet recruiting, with another preparation, another pharmaceutical company. Okay, so this is these are these preliminary results of the sarcoglycan gene therapy study, which demonstrate no sarcoglycan expression before the therapy and sarcoglycan expression after the gene injection. Okay. Apart from gene therapies, there is another, a very simple particle, very simple substance, which is tested in limb-girdle muscular dystrophy type 9, R9. This is ribitol. Ribitol, this is a very simple chemical substance, which is a sugar alcohol formed by the reduction of ribose, and it is a substrate for dystroglycan glycosylation by FKRP. This is the enzyme which is mutated in this type of limb-girdle muscular dystrophy. Dystroglycan is present in the muscle cell membrane, and it is very highly glycosylated. It means that there are sugars which are connected to the protein core, and one of the enzymes which connects these sugars is fukutin-related protein. These sugars connect the dystroglycan and muscle cell membrane to the extracellular matrix, to the laminin here. And in normal situation, when FKRP is not mutated, it connects the ribitol to the alpha dystroglycan, and this glycosylates alpha dystroglycan and connects it to laminin. Mutant FKRP is unable to do it. This activity is only trace here, and only some ribitol particles are incorporated into the glycosylation process. But in a situation of exogenous ribitol excess, this mutated FKRP protein, this mutated enzyme, which has an excess of the substrates, starts to work better, more efficiently, and glycosylates alpha dystroglycan more efficiently. This was proved in preclinical models, and now clinical trials have started. Phase 2 trial is ongoing, not recruiting the patients currently, but it has very encouraging preliminary results as well. This is the dystroglycan glycosylation level pre-therapy and post-therapy, day 19. And we can see that the therapy is the reduction of the creatine kinase level. Okay, sorry, I must. This is these are the results of 10-minute walking tests. Phase 3 study is planned soon, possibly this year. Another type of muscular dystrophy, facioscapulohumeral dystrophy. Here we have also some very promising preliminary data. A very interesting drug which is tested is losmapimod by Fulcrum Therapeutics. It is an inhibitor of P38 MAP kinase. This inhibitor reduced DUX activity in clinical models. DUX, it is a gene which is overactive in the fascioscapulohumeral dystrophy. So a phase 2 study, REDUX-4, was constructed to show whether this losmapimod reduces DUX and improves the clinical parameters in patients with fascioscapulohumeral dystrophy. Unfortunately, the results show no DUX4 reduction in the patients, but better functional outcomes. For example, this is the reachable workspace. This is the distance, the space which the patient can reach by his hand, because this kind of muscular dystrophy predominantly affects upper limbs. Yeah, so this reachable workspace was better in patients treated by the drug than with placebo. Muscle strength tested by dynamometry was also better in patients treated with the drug. And now there is a phase 3 study called REACH, which is recruiting the patients not only in the USA, but also there are some centers in Europe which are involved in this study. And I hope that the results will be as optimistic as the preliminary results. Another trials in fascioscapulohumeral dystrophy are trials with creatine monohydrate. This is a dietary supplement with anti-myostatin antibody. Let's hope that this will give promising results, because up to now, anti-myostatin antibody, we had great hopes, but the results were disappointing a bit in Duchenne dystrophy, for example. And trial with antioxidants used in Montpellier in France. And the last type of muscular dystrophy, which I would like to present, is myotonic dystrophy. It is also a very common type of muscular dystrophy, the most common one in adult patients worldwide. In this type of muscular dystrophy, the group of drugs which are of particular interest lately are antibody-bound antisense oligonucleotides or small inhibitory RNA, which are blocking the DMP kinase. DMP kinase, DMPK, is a protein which is again overactive, and it's not overactive, but it forms a large amount of mutated abnormal mRNA, which is toxic for in the long term for the cell, which affects splicing of other proteins. So this abnormal DMPK is blocked by these oligonucleotides or small inhibitory RNA. And there are two clinical trials by different pharma companies. First is Marina study with siRNA antibody conjugated, and the other one is ACHIEVE trial, which is now recruiting the patient, but only in New Zealand. Very similar mechanism of action, and we will see whether the results will reflect the very encouraging preclinical data for these two substances. Another drug which was tested in a very specific type of myotonic dystrophy, namely congenital type, a very severe form of myotonic dystrophy, congenital or juvenile type, is tideglutide. Tideglutide, it is an inhibitor of glycogen synthase kinase 3 beta. And the preliminary study found clinical benefit in the majority of subjects after 12 weeks of treatment. The results have been already published, and now there is a study called REACH, CDM, REACH, Congenital Dystrophy, Myotonic Dystrophy, which is recruiting the patients in some countries, unfortunately not in Europe yet. So as you can see, there is much going on in the clinical trials in different muscular dystrophies, not only in Duchenne one, which is I think it is a very optimistic for the future. In the contrary to the last 5-10 years, when we didn't have any option to these patients with muscular dystrophies. I would like finally to mention unfortunately, some most important negative clinical trials. Anti-myostatin antibody, as I already mentioned, disappointed us a bit. It was tested, for example, in LGMD-R9 FRP-related muscular dystrophy with no improvement. And moreover, there were some doubts about whether it may be impaired cardiac function. And another negative study, this is an old one, was deflazacort in dysferrin-related limb-girdle muscular dystrophy, which, in contrast to what was expected, caused decrease of muscle strength when the patient took the tested drug. And another drug which was which raised high hopes a few years ago. It is Resolaris. My patients in Poland always ask me about this drug. Is it in the market already? I don't know. After some promising phase 1-2 data, these are the number of clinical trials, there were plenty of them. This drug was granted fast track designation and orphan drug designation in 1915-1917, but then it simply disappeared from the company's pipeline, and probably there are no new trials with this drug. Okay, so thank you very much. I hope that this brief review was interesting for the patients and doctor community. And I would like to wish you all happy holiday season. And again, thank you for inviting me.
Thank you very much for this presentation. And I would like to open the floor for questions and comments.
Hello, hi. I have a question about the very last drug. When the company presents at any kind of conferences, do you, as researchers, ask about what happened to this, why the results are not published, why has it disappeared, and why there's no information for the patient community?
This is a very difficult question, because this company now in her pipeline does not have any drug tested for any neuromuscular condition. Yeah, they focused on other clinical areas, so we do not have contact with the company representatives in the conferences. Yes, I am puzzled what happened to this drug, because the first clinical data were quite optimistic, although there were some adverse effects, and maybe that's it. Okay.
Thank you.
Mm-hmm.